Hi, we're reporting from the National Center for Biological Sciences, which is a beautiful campus in Bangalore. And today we are here to visit Shannon Olsson's laboratory, which works on insects. And before going into what she exactly does, I just give you an introduction. So, how do I identify objects in my environment? Using my eyes, I can know if that's a tree. If a wild animal is after me, with my eyes I can identify it and I can run away from it. Using my nose, using odor, I can smell good food and I can go towards it and eat it. And if something smells bad, I can go away from it and not eat it. It might be harmful for me. Using hearing, my friends can call me, and that's how I can turn around and look at them. And if a friend of mine is telling me, there's something really bad happening, run away from here, that's how I can avoid danger. So identifying objects in my environment is extremely important. My survival is crucial. And the thing is, most of this object identification I've learned from my parents. So when I was a kid, my dad told me not to talk to strangers, not to take food from strangers. My mom told me what to eat and what not to eat. Languages I learned at school. Imagine the similar thing for an insect. An insect comes out of its pupal case, and the moment it's out, it has to decide everything in the environment, from what to eat, what not to eat, where to go, who to avoid, what predator to avoid, and it has no mother and father, it has no training. Now the world of insects is really different. They're extremely small and most of them are flying at extremely high speeds. So the world around them is rapidly changing. The kind of sensory organs they have are quite different. The organs they have are adapted to their environment. It helps them to identify objects in their own environment. And along with that, they have a teeny tiny sized brain which is processing all this information from the environment, the odor, the vision references. So today we are here at Shannon Olsson's lab to exactly find out how insects are identifying objects in their environment, and we are going to understand that not just at a behavioral level but also from a sensory perspective and from a neurobiological perspective. So without further ado, let me just take you to Shannon Olsson's lab. Let's head there. Come. So her lab is called NICE, which basically stands for Naturalist Inspired Chemical Ecology, and what that exactly means, let's go ask from Shannon herself. So it's Shannon out there, you've labeled her up. Hi Shannon.
Hello.
Have a seat.
Hello.
So what do you guys do out here?
So we primarily study how animals know what things are in the world around them, just like we have to know. And you probably already explained this a bit.
Yeah.
But it's a really fundamental idea for all of life to be able to know the important things they need to survive. They need to know what they should eat, what they should not eat, what they should run towards or run away from. They also need to, most of the time, find mates if they're a sexually reproducing species, like most organisms are.
Yeah.
And this is true for all animals on the planet. And we particularly are interested in insects because, well, for one, they are incredibly diverse.
Right.
They are some of the most ancient animals on the planet. They're nearly 500 million years old.
Wow.
And half a billion years old. Yeah, they're some of the first animals to have conquered the land, actually. They're really, really old. And there also are nearly as many species of insects on the planet as there are all other organisms put together. So if you count, vertebrates, so all of us, fish, birds, everything, lizards, and also the invertebrates and also even microbes and even plants, all that put together is just about equal to the amount of insects on the planet. So
Wow.
they are really, really successful. Yeah, and the part of the reason they're successful is because they're really good at identifying things in their environment and adapting to them. So that's why one of the reasons that we study what we do.
Great. So can you tell us more about how the world of an insect is different from my world?
So one of the primary things that separates most insects, not all, there are some social insects like bees, but a lot of insects are non-social, which means they don't have a home and they don't have anyone to learn from. So when they come out from their egg or from their pupal case or their chrysalis or whatever you want to call it, they are by themselves and everyone around them is equally ignorant of the world as they are. So they have to have some ability to find things immediately, like food, for example. Obviously, if they have no idea what food is, then they're going to die pretty quickly. And if they have no idea what danger is, then they're also probably going to be killed pretty quickly. So unlike us who mostly learn about things around us, we do have some ability to innately recognize some things. Faces are one of the things that scientists believe that humans can recognize even at birth. But for insects, they definitely have to recognize a lot more things, and that's what makes them somewhat different than us. And that also means that they have actually in their brains some sort of template for recognizing their food, for example. So if you're a butterfly and your main food is nectar from flowers, well then most likely you have some very loose representation of a flower object in your brain, because otherwise you'd be landing on every rock and tree and soil and maybe cars and houses, and you'd probably be dead before you find the right object. So this is one of the things that really separates them from us. And also they have really tiny brains. Obviously, that's one of the more obvious things that I think most people would know. They have only roughly around 200,000 to 500,000 neurons in their brain, and we have 80 billion. So obviously the level of processing, the level of thinking that they can do is obviously a lot less, but yet they still do pretty complicated things.
What's your approach for studying insects? Right outside, I read that your lab is called NICE Lab.
Yeah.
And that stands for Naturalist Inspired Chemical Ecology.
Yes.
So I sort of got an idea, but I would like to know more.
Yeah, we try to be nice is one, but it's besides being a pun, it's also a serious name. So naturalist inspired means that not that we gain inspiration from nature, which I hope most biologists do to some extent, but actually a naturalist is a very specific type of scientist. So a naturalist is a scientist who actually goes out into the world and studies animals for their own sake. They're not necessarily studying them because they're going to cure cancer or because they're going to provide them some insight on a particular cell or nervous system, but they're actually trying to understand what makes that animal tick. How does it work, and how does it work in its natural environment? So that's a really different way of thinking than a lab-based analysis would be, where you take the animal into the lab and look at it. Now that said, we actually, while we take the information from nature and we try to start as much as possible in the natural world, so we do a lot of field research. We look at our animals in the Himalayas or in Coorg or in Kashmir or wherever they are living. We look at them there. We do bring them into the lab. And so one of the things that we're most interested in is the chemical ecology. That's the CE part of the NICE name. And that is because insects, while they have a lot of the same senses we do, they can touch and taste and hear and smell, they actually don't use these senses to the same extent that we do. Humans are primarily visual, right? Most of what we get from our world is by seeing it first, like a mango. Like if you want to get a mango, the first thing you do is look for something that looks like a mango. Then you'll pick it up and maybe squeeze it, see if it's ripe, and then you'll smell it. But insects are probably the other way around. They probably smell the mango way before they actually really pay attention to how it looks. Then they see it, and then they probably land on it and get the taste and the touch. So it's a little bit reversed, but that means that the odors, the chemicals, are actually a really primary means by which they identify things. So the naturalist inspired chemical ecology allows us to understand how insects identify objects in the natural world and then try to understand how they perceive those objects using their nervous system, their brain.
Right. So tell me something about your background. How did you get into all of this? You must be a chemist, a neurobiologist, and you've studied evolutionary biology. So I want to know more about it.
Yeah, maybe I'm a jack of all trades and master of none, I don't know. But my background, my bachelor's degree is in chemistry. And actually, I've always been fascinated by science, actually. I really liked science from the time I was a very small child. I grew up in a very rural area of the U.S. in upstate New York, but I spent a lot of my childhood outside, and I had really amazing parents who encouraged me to pursue anything I was interested in. So I remember once my mother took me to the local hospital and took me to the lab there and asked for a petri dish, and I did the whole touching the petri dish and watching what grows to get an idea of what bacteria looked like and fungi looked like. And I think I was maybe 7 or 8 years old. So my parents were great that way, and they didn't necessarily send me to the best schools or anything like that, but they really encouraged me to explore the world around me, and that passion was really instilled at a very young age for me. And I also had great teachers who encouraged me. I went to a camp actually when I was about 13, and it was a camp for women in science. And it was the very first time that I had met a scientist who was a woman, because most scientists that I'd seen were men, and most scientists in my history books were men, and with the exception of women like Marie Curie and a few others, it was mostly men. So I actually didn't get a sense that I could be a scientist until I went to this camp, and I met all these amazing women who were doing all these cool things with DNA and cells and making new chemicals and making plastics and things like that. And I thought, wow, I can really maybe do this as well. So these very early experiences really inspired me, and I think that they're incredibly important. And it's one of the things I retain to this day, that it's very important to inspire children at a young age. So it's also something that I'm very adamant about here in India as well.
Right. And what got you to India? You were telling me something about the biodiversity of insects out here.
Yes. So why am I in India? It's like probably the most common question I get asked, because I'm not Indian. My husband's Swedish, and we have no family here. We have now friends, but we didn't before we came. And there's really 3 reasons. It's very simple. One is the biodiversity. 17% of the world's biodiversity hotspots are in this country. It's incredibly diverse. There's lots of ecosystems, desert and rainforest, and Himalayas, the highest mountains in the world, and oceans. All sorts of everything you want is here. And it also has 20% of the world's population, and it's going to be the most populous country in the world in about 10 years, which means that it not only has a lot of possibilities, but a lot of problems. And I think that is both a challenge and an opportunity for us scientists to try to understand better how a growing population, a growing world population, can actually live with the natural world. So some of the things that we research, while it sounds kind of like, oh, you're interested in how insects identify objects, well, that's very cute, but that's not very useful. It is extremely useful, because insects not only are things that cause disease, malaria, dengue, which they do by identifying people, they destroy our crops, they also are pollinators. Nearly 80% of our plant crop species are pollinated by insects and animals. And so actually understanding that's very important for our food. And they also get rid of the waste on this planet. Like termites have termite mounds, and they actually get rid of a lot of the organic waste in the soil that actually keeps us from having tons of garbage laying around. So they're really important. So actually by studying these things, and actually by extrapolating from insects how they identify objects, we can also think about other organisms as well. So that's really a broader goal that we have, is to really understand the natural world and be able to live with it.
Can you show me some of the experiments that you're doing in the lab?
I'd be happy to.
Okay,
so I'll try to take you, the one experiment I can show you is actually out there. Right? So obviously that is the most important thing. So I hope that whoever is listening to this will think that all of our research really is inspired by what's going outside. So I hope that whoever is listening will, at the end of this, go outside and go and look for an insect, look at an ant, look at a grasshopper, find something, find even a housefly, and just think a little bit about what they're doing, and maybe think about how what they're doing is maybe not so fundamentally different than what we do as well. Okay, so that's the first step.
Great.
Okay. So this is a bit of a noisy lab, so I'll talk a little bit louder than I normally do. I mentioned that insects generally use smell as one of the first ways to identify objects. They certainly see, they have amazing eyes actually, as other people on our campus work on, but they also, smell is very, very important to them. So that means we actually, in order to understand the objects that they identify in nature, we have to be able to identify what they smell and the smells of those objects. So this is a chemical analysis facility. This is actually an NCBS facility that's actually technically separate from our lab, but we maintain the facility. And we actually do research for people all over India. There's a couple of excellent women scientists here. They're coming from UAS right next door, and they're doing a research on some fungi that live inside of plants. They're called endophytes, and they're extremely important for the plant to survive. And they give the plant a lot of qualities that allow it to actually survive in nature, so they're working here as well. And what they're doing, if you look here, she's actually preparing it. She has a petri dish with this fungi growing on it.
Oh.
And she's actually going to be able to take a fiber, which you can see here, and this fiber has an adsorbent on it. Adsorbent means it attracts chemicals to it. And this adsorbent surface will attract all the chemicals, all the smells that are coming from that fungus. We can do the same thing with a tree leaf or a flower or a fruit. And we can even do this outside in nature, which is what I said, most of our work starts out in nature. And once we've collected those smells on this little fiber, we can actually stick it directly into this instrument. This is called a gas chromatograph. A gas chromatograph is an instrument that separates chemicals into their components. You may know that anything that we smell is actually made up of many different kinds of molecules. So when you smell your morning coffee or you eat your chapati, that's actually, you're smelling and tasting not just one molecule, but many of them. So in order to identify the smells, we have to be able to separate them one by one. This machine is what allows us to do this. And then we have something here called a mass spectrometer, which allows us to identify, it's like it gives us a fingerprint of what the chemicals are. So this device is incredibly important for us to be able to identify what the objects are. What is it that the insect would actually be detecting in its environment? Maybe I also show you what Abhinav is doing. So Abhinav has here a bee, a honeybee actually. So this honeybee actually was collected right outside on our campus just a couple of hours ago. And what Abhinav is trying to understand is how that insect detects those chemicals that we just identified. So once we've identified those smells of the flower, in this case, because the bees like flowers, they're pollinators, then he actually can present them with these smells and look at how their antenna, which is the insect nose is the antenna, how the insect nose detects these different chemicals. So in each of these different tubes right here, he can actually pass air through it, and it's a way of presenting these smells one by one to the insect. And that gives us an idea of how they actually detect these chemicals and how they smell them. What he has over here, I'll move out of the way so you can see it, is actually the electrical signal that's coming from the antenna. So they have neurons just like we do. They just have fewer of them, and they're of course quite small, but they also have neurons, and you can see this is the electrical activity. It's a bit noisy because he's just trying to get a contact right now. But this fluctuation is fluctuations in the electricity between his electrode and the animal.
So basically, there's an insect which has been mounted there, and you have electrodes inside the antenna of the insects.
Yeah, so a lot of people ask us if the insect's alive, and the answer is absolutely, because it will not respond to things when it's dead. So the insect is completely alive. It's actually restrained in a little pipette tip just so it doesn't fly away. We of course can't really hurt the insect. If we hurt it very badly, then it's also not going to respond to us. So we try to do as minimal damage as we can to the animal, and then we look at how it responds to odors. So now you can see the signal is leveling out a bit on the screen.
So how difficult or easy it is to get a small electrode inside the antenna of an insect?
It is actually quite difficult. There's a lot of things you have to have working. I mean, you have to have a very steady hand. I always actually have to drink a cup of coffee before I mount the insects because I'm a caffeine addict, so it actually keeps me calmer. But I had this lovely, very famous German scientist tell me that, you know, just a little bit of beer or whiskey before you start is actually a good way of, you know, relaxing. I don't necessarily, you know, suggest this to my students, but that's what he did. So I think everybody has their own method to calm down, to relax. You have to make sure that the animal is in good condition. You don't want to be too rough with it. You have to be kind to the animal because you actually want it to survive and live and give you answers. And we can't talk to insects, right? We have no way of asking them, did you smell that? So this is actually our way of asking them a question, right? And the only way that they can answer for us, for our language, is by detecting how their activity of their body, what it tells us. So this is our way of.
So in terms of size, how small is that one cell in which you're inserting a needle?
So the brain of a standard insect is roughly 1/2 to 1 millimeter in size. The whole brain fits into a very small area, and they have about 200,000 to 500,000 neurons in their brain, as opposed to us, which have 80 billion neurons. So it's pretty small. The size of the single neurons on the antenna and the area that we have to connect with is about 1 micrometer. So it's very, very small.
Wow.
1/1,000,000th of a meter, right?
So I remember you telling me something about your grad days and how you coped up with electrophysiology.
Yeah, so all of the techniques in this lab are techniques, with the exception of one that we'll see a little bit later, techniques that I have done during my own career. So I struggled with them the same as my students, which is actually good because I often say that my job as a PI is actually just that I've made the mistakes so they don't have to. So that's really a lot of what I do, is just tell them what not to do because I've done it all wrong. And one of the days, I worked at a place called the New York State Agricultural Experiment Station, which is part of Cornell University, which is where I did my PhD. And one day we had Hillary Clinton's team, because she was Senator of New York at the time, coming to visit our labs and see what we do. And I had everything prepared, just like Abhinav is doing right here, everything set up, and nothing worked. And I was so frustrated. It worked all the time before they came, but of course things got delayed, and by the time they came, the animal was pretty much dead. Nothing was working. And so I worked really hard and I got everything set up again. And as they were walking to the car, I said, no, you have to come back, it's working, it's working. So I dragged them back in, and they were really kind and you know, excited, because I was so excited by it. So so I think that this is also very important, what you guys are doing to really show science to people who are not scientists and may not see what it takes. So.
I'm Abhinav. I just finished I'm an intern at Shannon's lab. I just finished my undergrad in chemistry from the Shiv Nadar University. It's near Delhi, which is where I was brought up. And yeah, so I did a 4-year undergrad in which the final year was basically research oriented, and a lot of my work, I mean, a lot of the research that I did was associated with physical chemistry. It was all photophysical chemistry was the kind of stuff I did. And however, a lot of the courses that I took out of interest were also associated with biology and, you know, neurosciences and stuff like that, which is how basically I got interested in the kind of work that Shannon does. we basically take bees from 4 different locations in Bangalore that have different levels of pollution, and what I do is I check their abilities to detect odor depending on where they are from, and how much possibly the pollution or where they are from is affecting their ability to detect odor. And I'll show you how this device works. It's basically what you do is you take one of these capillary tubes from here, and what it basically does is it takes one of these capillary tubes. You can see it's really thin already, but it has to be a lot thinner because of the size of the insect's antenna. And just a second. Any second now. It'll basically what it basically does is it takes one of these really thin capillaries and pulls it on both ends after heating it, and it gives you these 2 really thin tips to the electrodes. And I don't know if you can see it. The tip of the electrode is now really thin, and what that in turn does is that when you stick an electrical wire into it and put it into an electrical holder, you stick one of these into the eye, one of these tips, and the other one onto the antenna, and that's what causes the circuit to complete. And every time you pass an odor over the antenna, you can see an actual dip in a spike in the spectrum, in the electroantennogram, and that's how you notice that the insect is actually detecting odor, which is what I do. So yeah.
All right. So where are we going next?
Yeah, so we started outside, right, which unfortunately I couldn't show you, but you can imagine it. And you can go outside and actually see it for yourself and watch insects, which is basically what we do. I took you to the chemistry lab to see how we identify the smells and things that they detect on these objects. You saw how we look at the electrical activity on the antenna, but those neurons on the antenna go to the brain.
The brain, yeah.
Yeah, so of course that's where the magic happens. That's where the animals actually identify what things are. They use the brain. They have a brain just like we do. They have places for memory and learning. They have places for locomotion. They have places for detecting visual cues or chemical cues. And so where we go next is actually trying to understand their brain. So I'll take you in here.
I'm really excited.
That is the teeny tiny sized brain with the and she just took it out in about 5 minutes, less than 5 minutes,
that
right?
Processes
yeah
all the information.
Yes,
in
that
an
is it.
insect's
That
life.
is where all the magic happens in that tiny little thing, and it looks a little bit.
That is
if
fascinating.
she faces it, now she's got it. It's perfect. Good job, Janelle. So you see these 2 sort of big things on the side. It looks a little bit like a Batman symbol. So this is actually the optic lobes of the fly. So these 2 things on the side are where they process visual information. And the center part, which looks a little bit like a heart when she's got it. Oh, this is fine. So you can see the center looks a little bit like a heart. That's actually where all the olfaction and learning and memory and coordination, all that happens. So the 2 wings of the Batman are actually just for vision. So certainly they do detect visual cues. It's not just odors. They definitely see things, and you can see they have a huge amount of their brain devoted to just seeing. So that's what the brain looks like.
Great.
Maybe I'll show you what's going on over here. So if you can take a shot of this right here and out. This is actually the heart of a bee. So this is how the bee circulates throughout its body. It works the same as our heart, but it looks very different. It's actually a long channel. And what Gita is doing is Gita's working on a project on the effect of pollution on bees. And so she's collecting bees from all different areas of Bangalore, from polluted areas and less polluted areas, and she's looking at their cardiac function, just like we would look for heartbeat or heart murmurs or heart arrhythmia in a human being and look for evidence of malfunction in your heart we're doing the same thing for these bees. We're looking at evidence for cardiac function, which it suggests that there's changes there. We're looking at levels of living blood cells in their body. We're also looking at how they smell and how maybe the levels of pollution may change their ability to smell, and maybe even their ability to locate flowers. And this is of course a really big problem because 10 of the 20 most polluted cities in the world currently are in this country. And most of those cities are not, you know, Delhi-sized or Bangalore-sized cities, but they're actually second-tier cities, which are really close to agricultural lands. And a lot of these bees move around, which means they'll move in and out of the city. And if this pollution is affecting them as much as we think it has, it will also affect us as well, because it's affecting our ability to pollinate our crops. So this is one of the things we're working on as well.
It's just beautiful how the heart of the insect seems to be beating.
Yeah, it's really amazing to watch it. And Gita does this really, again, you know, this insect has to be alive, right? I mean, we can't, obviously you don't have a heartbeat from a dead organism, so she has to be incredibly delicate in how she dissects these bees and opens them up to be able to see their heart rate and their heart function. And so she's looking at this from bees from very polluted areas, where like the industrial areas of Bangalore, which is like Pina, and also very rural areas outside of Bangalore as a comparison to see how pollution might be affecting them.
So you're measuring heart rate, and what else?
We're measuring heart rate, we're measuring blood cell count, we're measuring their behavior outside. As I said, we always do our work outside. She's doing scanning microscopy of the surface of the bee, where you can actually see the pollution on the surface of the bee's body along with the pollen. She is even doing some genomic analyses to look at how the different proteins in their body are changing, whether they're in a polluted or a less polluted environment. So she's basically, everything she comes up with is all from her. She's really an amazing scientist. So.
And how long can you keep the bee alive like that?
For at least 15 minutes, because our measurements actually take at least 15 minutes long. There's like 3 parts to the heart. There's basically an upper, a middle, and a lower part, and she actually has to image each of those and the heartbeats for 5 minutes each, so it has to be alive for at least 15 minutes, and it can stay alive for much longer. It actually depends on the bee, right? And that's one of the things that we're paying attention to, is how long they do stay alive. So
great. Amazing.
Okay.
Never seen an insect heart beating before.
Hi, I'm Hinal. I'm a PhD student in Shannon's lab. Currently, I'm working with the project on a fruit fly. How I got into this lab is a very interesting story. I did my master's in a university called Maharaja Sayajirao University of Baroda. I studied zoology. I have worked with birds before, and I realized studying birds, there are 2 important species on this earth, is insects and birds. And I heard Shannon giving a talk once, and that was so fascinating to me, how these amazing tiny insects are. And they have a very interesting going on in their brain. So all I wanted to do is how these tiny animals respond when we give them some odors. And this is what currently I am working on. I am looking at their brains and try to identify there are chemicals which are present and which are regulating their day and night behavior. And these chemicals are produced in very tiny amount, but they have really major effects on insects' life. So currently you can see here, like I have dissected the fly brain, and this is the area where I am interested, because this is where the next level of processing happens in the brain. And I am not sure if you can see it, but this is where I am interested in, and I am looking at the chemicals which are present in this tiny brain part called enteral lobe, where the smell is further getting processed in their brain.
Can you explain this instrument here?
This is called a stereomicroscope. We use this to visualize the specimens which are really tiny, which we cannot see through naked eye, and this is really helpful for me to see a particular region, because it's a very tiny part. I think it's few microns, so it helps me to identify the region so that I can just pick up that part in my fly brain.
And finally, can you tell me a little bit about what you see the world of flies and insects as? Like, what are they?
I think after joining this lab, I realized that insects are really important, and they are actually telling us more about how the surrounding, like their presence makes me that how this environment is currently, like we can see that as global warming is going on, and I can see this insect are reducing in their numbers, and we are not able to find them in nature, which is a sign that we should start saving insect.
Anything else you want to say?
Anything else I want to say? So when I sit in my room and I see insect, I used to get scared before, and all I want to do is just kill them or just shoo away them from my home. But after reading more about them, I realized that they are really important and we should really care about them. Yeah. So I started working with insect first in Axel's lab, where I used to work on honeybees. And you might know how important honeybees are. They are the pollinators which are important for almonds and other fruits. Without them, we might not get many crops. So they are really ecologically and economically important. So that's where my work on insects started. And then I started reading more about insects, and then I met Shannon, and then I realized I think this is what I want to do for my research, and I continued as a PhD student here.
Especially flies is different from how we grow up, right?
Yes.
How do flies grow up?
So there's two types of insects. There is something called a hemimetabolous and a holometabolous, and those are very big words that actually indicate how they develop. So a hemimetabolous insect is a little bit closer to what we do. We also start out as an egg, and then they go through something called a nymphal stage, which is it sort of looks a little bit different than the adult, but it still has some of similar stages, and then they become an adult, like a praying mantis does this. There's also something called a holometabolous insect, which is the butterfly that goes through metamorphosis. So it completely changes its body. In fact, not even the same tissues are around as when it's a baby or a caterpillar or a larvae as it is an adult. And usually they don't fly when they're in this juvenile stage as well. So that makes them, as you noted, very, very different than us, because they go through different stages. So we have in here, this is where we keep our insects. This is a somewhat climate controlled. India's great, actually. Unlike where I used to live and work in Europe and in North America, I didn't have to do climate control like I did there when it was, 0 or below 0. But even here we still try to keep the humidity up. Like in the summertime here, it's very dry, and we need to keep the temperature fairly regulated. We don't want it to get too hot or too cold, and most importantly, we regulate the light-dark cycle. The Nobel Prize, as your group just talked about, the circadian rhythm, actually, this is incredibly important. It's also, as you mentioned, it's so important to everything, and as biologists, we have to pay attention to this. So our insects also, we have to make sure that their rhythms of their body are in sync, so we also regulate that with the light-dark cycle. So this is what it looks like. I'll just stick in here so you can come in too. So these are all different kinds of flies in here. So these are called hoverflies. So hoverflies it's actually another project we're doing. Hoverflies are amazing pollinators. They are actually cosmopolitan, many of them, which means they're on every continent except for Antarctica. And they're not as good as bees. Bees are really good pollinators. They have pollen that they carry in sacs on their bodies. But they are perhaps the most numerous pollinators on the earth, because they can live in so many different environments. So we're studying them and looking at how they identify flowers in different places in the world. So this is work we're doing with Karin Nordstrom. She's a researcher at Flinders University in Adelaide. So these down here are apple flies. So, as the name implies, they really like apples, and that was the brain that you would have seen dissected. It's actually an apple fly, and they're single-minded. They only like apples. So that's called a specialist, which, as the name implies, they are specialized only on one thing, which is apples. And we actually are getting these as pupae, not as eggs. So we're getting them as pupae from the ground and having them emerge out here. in the lab. So this is where our insects live, if we're not collecting them from outside, which we also do.
Great. Where do we go next?
Okay.
Yeah?
Now you've seen the beginning, and you've seen the brain, and you've seen the heart. Now this is actually where we find out what they're actually thinking about. So what we're doing here is actually neurophysiology, which means we're trying to understand how the neurons of the insect's brain actually are working. So Cheyenne actually has an apple fly, which is the species I just showed you, and apple flies, I told you, are single-minded, so they only like apples. And Cheyenne is actually, can you hear that? I'll get closer. Can you hear that?
That's the brain beating.
That is the sound of a single neuron in the brain firing. So neurons carry electrical signals, and the type of those signals and the number of those signals is how it carries information, and carries information throughout the brain. And that's what she's detecting right now, is the sound of a single neuron. And this is what it would look like when it's responding to apple smell in this case. So what Cheyenne is able to do is actually, once she opens up the fly, and again the fly has to be alive because you don't think very well when you're dead. So these are living insects that she's very, very carefully just done a little bit of brain surgery on them just to open up the brain so she can get to it. And she has a very tiny electrode that she sticks very deep into the brain so she can get to where they're detecting odors. And that's what you can see right here. Here's her preparation.
So how do you manage to get inside the brain of an insect?
Well, as I said, they have an exoskeleton. So they have this thing called a cuticle, which you can open up just on the top of their heads. And you just like a brain surgeon would do surgery on a human brain, you have to cut a little window in the skull, and there's the brain. And that's basically, we do the same thing, just with much smaller tools than you would on a human. And Cheyenne is really good at this. She's actually become an expert. And she actually comes from the University of Notre Dame. So she has a completely separate aspect to this project, which is on population genetics. She works with Jeff Vader's group there, and here she's doing neuroscience. So it's another example of how our research really goes through a lot of disciplines. So that's what she's doing.
And she gives it different kind of odors?
Yep, apple smells, right? Because they're an apple fly. That's what they like. And through a lot of analyses, including field work that we've done, we know that's what they like. So we already know what they like, and now we're trying to figure out how they know that they like it, basically.
So you're literally getting inside their heads.
We're literally trying to get inside their heads, yeah. And again, that's the best way we have. We have no way of talking to them, right? So only by doing these kind of analyses do we really get an idea of what they're thinking.
So, hi, my name is Cheyenne Tate. I am a 6-year PhD student in the biological sciences at the University of Notre Dame. So I got into biology because I've always been interested in the natural world. I grew up in rural New York on a dairy farm and was always running around as a kid catching bugs and lizards and frogs and things like that. So animals and nature have always been a big part of my life, but as I went into college, I was wondering more how everything worked, and I got sort of into evolutionary biology actually. And then when I started graduate school, getting into the mechanisms of evolution by looking at population genetics, which is actually my major field of study at the University of Notre Dame, where I'm a graduate student, and actually then neurobiological mechanisms as well, which are what the genetics of these specific flies I study are altering the neurobiology of the flies so that they make different decisions about their host plants. So that's sort of a little bit of who I am and what I do as a graduate student. More specifically, what I've done is a lot of neurobiological dissections of these flies and measuring their neural responses to different odors of their host plants at different levels. So from the outside in, so the antenna where they pick up the raw signals from nature, working up the neurobiological pathways into the brain itself. So doing a lot of micro dissections and figuring out the best angles and how to get at the brain with the electrodes and various tools that we use to get the strongest signals, the clearest signals, to actually start to figure out how these neural pathways add up to behavior and can change the course of their evolution. So that's what I do in science.
Okay.
So a question I actually get a lot with my relatives who are not really in the sciences is, so flies and insects, they have brains? I'm like, yes, of course they have brains. They have to make sense of the world around them and make really simple decisions, but they still make decisions in order to be fit in the biological world. And also, I also get the question a lot of why do I do what I do and why is it important? I'm studying one species of insect, one neural pathway in that insect that may or may not have larger cascading effects on that insect, but still it's only one neural pathway in one insect. And well, the apple maggot fly, which I study, is an agricultural pest. It's not something like a mosquito that might carry diseases that impact humans. Instead, it's just this fruit fly that flies around and does what it does. Why am I studying that? So I think a lot of science is being curious about things like this that other people might not have the time or energy to be curious about, and fostering curiosity, because you never really know what you might uncover, and since science is always building on itself, if it might lead to really important discoveries decades down the road. So those are just questions I get from relatives and friends and normal people in the field, or outreach activities like, why is important and why am I doing what I'm doing and not something else that's potentially more helpful. But we can't all do things that are directly applicable in science, and curiosity drives you where it'll drive you. So this is what I'm most curious about in science.
And your first experience of India and what it was like, and yeah.
Okay, all right. So I started working with Shannon when she was still in Germany. So my advisor back in the US had been to Germany before, and that's what when he was selling Shannon to me, he was sort of selling Germany to me. But in the course of working with Shannon, she got recruited and came to NCBS in Bangalore as a PI, and then was like, and then the way my project was going, we knew I would have to follow Shannon where she was going so I could finish my project. So we knew I would be coming to Bangalore long before I actually made it here. And of course, I was a little bit apprehensive about coming because I'd never been to India before. I'd never been outside of the US and Europe. But it's been really great here at NCBS. So I come from a large research university with a lot of undergrads. NCBS is it of course has no undergrads, and it's a research institute of like the highest caliber. Like they have every toy I could want here in neurobiology. And I've also just liked being here because it's so collaborative. There are so many different labs working on neuroscience, even neuroscience insects like I'm working on, that I can get help when I'm confused on anything, which at my institute back in the US, or my university, not institute, back in the US, there's a few population genetics labs, but we all have different questions, so it's harder to say like your specific problem, get help on your specific problem. You reach to different universities and different collaborators, but here at NCBS it has really seemed like everyone is just here and can help you on things. So I've had a really good experience in India, and specifically here in NCBS, where I am a lot working on these these neuroscience techniques, where the one protocol takes like 14 hours the first day, and then you have consistent wash steps through the next 6 days. So I've been here at NCBS in this lab at this rig for many hours.
Final question, final question on the personal side, which is, how was your first day or first few days in India? First trip ever.
First few days.
Yeah.
Okay.
Did you think it was mad? Did you think what it was like?
All right. So I came to India for the first time in February 2015. And then I had a year break, and then I came back here in February 2017. So back in 2015, the first time I got here, I sort of stepped off the plane and was like, oh wow, it's really hot here in Bangalore. I got to the institute and talked to people who are from like Delhi in North India, and they're like, this is not hot at all, what are you talking about? I'm like, I am dying, I'm so sweating. The first week in India, I spent in the lab because it had, mostly because not I was working it was the first week, but because it had air conditioning. So that was one of the things I really had to get used to being here. I also think it was probably because I was coming from winter in New York State. So I was literally coming from like minus 20 degrees Celsius to like positive 32 degrees Celsius. So it was like a huge difference in climate. So it's kind of a bad year to come because for us, or for me especially, it was a huge difference in climate, a huge difference in food, a huge difference in the crowds on the street. So I'm from upstate New York, and my town has less than 1,000 people, so I'm not from New York City. When I got here, I said maybe I should have gone to New York City more often to maybe just get used to a little bit of more people being around me all the time. I'm told even that wouldn't have prepared me for Bangalore level of traffic and crowds. But that was just another thing to get used to. And crossing the street, of course, was a major thing to get used to. People, I would be so apprehensive about it that someone would just grab my arm and pull me across the street here. They still do sometimes, but most of the time I follow now when I trust whoever I'm with to not like experiment. Has gone my experiments have gone many different ways and have eventually led me to recording the neurons in the brain. So originally I was trying to see larger brain activity of a whole region. So we did that with a different technique, which was calcium optical imaging. So using a dye to see activity in real time, to splotches of heat maps of activity. But that didn't work because it didn't work really well because my flies have a strange shaped brain, which is what I've been calling them lately. And they have to be tilted very far forward to see their brain from the top like you're looking at them. So when you tilt them forward, the region of the brain that I actually want to see, everything becomes on top of each other. All the regions are on top of each other, so you can't see differences in activity when they're all on top of each other. So we've switched techniques such that I'm poking a glass electrode into this specific region of the brain, but instead of seeing activity in the whole region now, I'm just looking at whatever single neuron I hit with my electrode. I'm testing it with various different odors to see how it responds, and based on the response, I'll know if it's a neuron that goes into the region I'm interested in or not. And so once I know I'm in the right neuron, and therefore a neuron that goes into the right region, I can stain it and take the brain out of the fly and use fluorophores to make that region glow, and then look at it under another microscope and see where that region is. So we're trying to get a map of where these specific regions are that respond to the odors that these flies are attracted to. And the main story is that there's different populations of the flies attracted to different host odors, and it's impacting their evolution. So we want to see how their brain maps differently to the odors. And the way we're doing this now is at a single neuron level, because the larger regional level didn't work out so well.
And can you explain the equipment we have in front of us?
Yep, I can explain this technique sort of as best I can. What I'm doing are intracellular recordings with this equipment around us. So I mount the fly upright on this mounting block here, so that its head is in a little cover slip, and the back of its head is exposed to me, so I can just cut a window in the skin on the back of the head and expose the brain, so that then when I look through the microscope I can see the brain. And when I have everything else set up around it, so when I have the electrodes set up around it, I can see the electrodes also through the objective, and then I can carefully manipulate the electrodes into the region of the brain that I'm interested in using this Scientifica, which just guides this robotic arm forward and backward, left and right, up and down, to get me exactly where I think I can see where I need to be in the fly's brain. And when you're in the brain, the noise level changes. So also I have this is controlling the signal coming out of the brain, and we can also push electricity and current into the brain using the amplifier. So reasons we'd want to push current into the brain is that sometimes you're not quite in a neuron, you can hear it and you feel you're starting to get close to it, but you're not quite in it. So if you buzz it, which is just a switch right here, if you buzz the current, you're pushing current through the glass electrode and you're just vibrating it slightly so that you get into whatever neuron it's next to that you can hear the spiking, but you know you're not quite into because it's not quite strong enough because you can see it as well. We're watching the spikes on the oscilloscope and on the computer screen at the same time. So, so you buzz current and you get the neuron spikes to spike strongly, and then you can test odors to it and you'll see the spikes will greatly tick up in activity level. And then when you're sure you're in the right neuron, it responds to the right odors. You push current back through it again because then the current pushes the dye into the cell and allows you to stain that whole neuron pathway.
Cool. Anything else you want to say?
I don't think so.
Yeah? Okay. Thank you.
So the last step in our lab anyways, of course we do a lot of collaborative research with other excellent scientists doing everything from engineering to population genetics, as I mentioned with Cheyenne's work, is once we know a little bit about what they're thinking, we actually want to try to really understand how they make those decisions in the world. So it's not only the fact that they have to identify objects, but they have to make a decision about those objects. Like if it's some food, they have to decide, oh, I want to go to that food. Or if it's something dangerous, oh, I need to go away from that. And since they don't necessarily have to do these things, it's not exactly like a reflex. It's really a choice that they're making. So they're using their brains to actually make a decision, really, not like we make decisions, obviously. They're not deciding where to go to college, but they do make very simple decisions: go or stay, fly or walk, go left, go right. The problem is when we're outside in nature, we can't manipulate it. So we can't just move trees around or make trees have fruit if they don't, or make trees get rid of fruit if they have it. So since we can't actually manipulate the world in reality, we do it with virtual reality, and that's something that Pavan has established in the lab. So this is an arena that we've created. If you've ever used virtual reality goggles or virtual reality glasses, you know what happens is you put these glasses on, and as you look around, the world moves. So you feel like you're actually looking around the Sistine Chapel, or the Leaning Tower of Pisa, or whatever it is. And we do the same thing for insects, but we can't put tiny little glasses on them, right? They're too small. So we actually build a world around them. So we have an arena that's three-dimensional. It's 360 degrees. It's two-dimensional, of course. It's not a real world, but it's 360 degrees because they can see in the back of their heads. And we have to give them smells because smell is very important, as I've been saying. And we have to give them wind because they have to feel like they're flying, for one. And secondly, when you smell something, it's traveling in the air and through the moving air. So the wind is what carries that smell to you. So they have to have the wind as well. So all three of these things have to come together. And then we put an insect in the middle. And you can see the arena. We'll show it close up so you can see it. And you can see them flying and then deciding. And what makes this special is that since it's all virtual, we can really dynamically change things. We can make trees disappear, or make fruit disappear, or give you fruit without a tree, or a tree without fruit. We can do all these manipulations to really get a sense not only of what they smell and how they smell it, but what decisions they make about it. And that's really what we're using this for. So this is the cockpit of Pavan's system. This is Pavan. He's a graduate student in the group. He's a Bangalore born and raised, and he developed this system in the lab along with some collaborators, but mostly by himself actually. This actually was helped by a researcher who came from Germany named Marian, who developed this graphical interface for the system as well, and also helped us develop the world. So I hope you can see here, this is actually a little insect, and this insect's not flying actually. This insect is just grooming grooming the wings right now. This is again an apple fly. So this is the same fly that Cheyenne is looking at in the brain. And it's on a ball right now, which is a sugar. A sugar ball. And what you hear now is the best way we have of knowing when they're smelling something, right? Because they are way more sensitive to smell than we are. So we can't actually smell when they're smelling. So the only way we know when we've given them odor is by having a sound produced when we produce the odor. So the sound is our proxy for knowing when they're getting an odor.
Getting an odor.
Getting an odor. Whether they're smelling it or not and responding to it is up to them. They have to make the decision. So if you can see, I don't know if you can come over here. I'll try to move the seat out of the way.
Yeah, you can.
So this is what the arena looks like. It's opened up right now so that you can see it. And you can see this tiny little insect right in the very center. And it's in the center of all these concentric tubes. And these tubes allow us to give it odor or wind from any direction. And right now the fly is not flying, but we're going to try to jump start it a little bit to get it to fly by puffing a little bit of air onto it.
Removing its ground.
Yeah. But you can see the system right now. So it's basically the insect is in a virtual world. It is of course not a completely realistic world, but it's enough that they actually will be able to make decisions in it. So Pavan's removing the ball right now. See, he's trying to take it so small. There we go. So now the insect is flying. And if you come back to the display, you can see it's shaking right now because he's just moved it, and now it's stabilizing. And you can see the insect flying. And these two markers right here are how we know where they want to go. You probably wondered, like, how do we know what they want to do when they're flying because they can't talk to us? Well, they're kind of like a swimmer would be, or if you're ever been in a rowboat or a kayak. When they want to go left, they will beat their wings harder on their right. When they want to go right, they beat their wings harder on their left. And that's how we know where they want to go. And we turn the world in response to their behavior.
The slightly misaligned, so that's what I shaped it.
So right now, Pavan is just fixing the offset for the fly. But you can see actually it's going right towards a tree. Yep. So you can see it actually going towards a tree. Now it decided to go towards this other tree. And it's really trying to get to those apples on the tree. But it can't land on the apples because there's no actual apples there on the tree to land on. So this is how we can manipulate their world and try to figure out what it is that actually makes them make decisions about objects. Is it the smell of the object? Is it the visual cue? How do those things play together? And
It's like a fly is playing a video game.
it is the fly playing a video game a bit, but it's actually not just really a fun thing to do, but it actually gives us real insight onto how their brains actually make decisions, which is the main purpose of this experiment.
Because they can also get where the
Yeah, so this fly, as you can see, is really, really working really well. It's making really great decisions about this tree and deciding to go in and around the branches. Now Pavan has taken away the tree, so now it has nothing to go to. So now it's just flying randomly in space. And this is a bit hard for us because this is completely unrealistic. There is never a place in the world where nothing is there. Right? There's always something on the earth. Even if you think in the desert, there's an undulation of the land. So this kind of landscape is completely foreign. But it gives us a sense of what they do in the absence of all objects, right? Which is probably a scary thought. I mean, if you were in a room or a world devoid of all objects. So that's kind of what we're placing them in now. So this is our baseline of what they do in this type of world.
And what do you exactly see? What happens when there's nothing in the world?
So they are a bit aimless, actually. They kind of go in random directions. One of the weirdest things that's recently happened is that when we gave them patterns in the sky of clouds, they actually seem to go to certain patterns in the clouds, and we're not really sure what this means, but it was quite bizarre and strange for us to discover this. But in the absence of anything to go to, they'll just kind of aimlessly wander. Whereas as you saw when we gave them a tree, they go to the tree. So yeah, so this is what it looks like when we give them clouds in the sky. And I don't really see anything in those clouds, and I don't think Pavan sees anything either, but the flies seem to see something. So they tend to actually go in very specific directions in the clouds. But always when there's a tree, they will prefer to go directly to the tree.
So if I understand it correctly, basically you're trying to figure out how insects find different things in their world, and on top of that you have control over the odor, you have
Yes.
control over what they
Yes,
see,
yes.
and you can change those things, and now you can figure out
Exactly.
how they respond to newer
Exactly.
things.
So this is kind of the culmination of all the other research put together, right? Once we have an idea of what they smell and what they see and how their brain looks and how their brain works, this is the very last step, which is actually trying to understand how they think and how they make decisions.
It's a very smart way of doing it.
Thank you. And thanks to Pavan, who actually built this as his PhD work.
I'm Pavan and I work at the NICE lab, and I try to understand how flies find food, and I do that by using virtual reality arenas. So we have built this virtual reality arena where we can understand what the fly is seeing, what the fly is smelling, what the fly is feeling. Partly why we have built all this is because it's just simply not possible to know what the fly is seeing, smelling and feeling at the same time in the real world, because I have no way of knowing a fly which is flying past there, what is it smelling there, because you never know. And moreover, I can never ever suddenly make a smell appear or smell disappear. And why this matters? Because almost all organisms find food, and they find food by using vision and olfaction, sort of, because let's say I ask you to find an apple. If I just say find a red round ball, that's clearly not good enough, because there's so many things which are red and round which are not apples. And let's say I tell you find the smell which smells fruity, and that's also clearly not enough, because there's so many things which smell fruity, even very similar to apples, like apples, pineapples and jackfruits. But if I say give me a red round ball which smells fruity on these smells, then there are not many things which are available. It's primarily an apple. And therefore you'll say naively, yes, vision and olfaction, combine them, life is nice. The problem is understanding them in reality is tricky, because you really don't know what they're doing in the real world, and you can't change them. And without changing, you never know what's happening, which is why we have this VR. So here you have a fly which you see here, and the fly is tethered on a needle, as you saw before. The fly is now flying, and you can see 16 pipes here, which is where it gets its odor and wind from. So I can control where the wind and the odor is coming from any of these pipes in real time. So I can make them smell like air is coming from 30 degrees left, or I can say odorous air is coming from 45 degrees right, or I can set any degrees like that. And also you have 3 monitors here which completely cover it in all directions, because they have eyes all over their head, and which will give them visual input. Visual input, something on these lines. So now what you see is basically a fly flying in the world and looking at the grass and the sky in the world. The problem with this arena is this: you don't know what the fly is doing. You don't know whether the fly is trying to turn left or trying to turn right, because they can't ask them, and they're not moving. They're stuck in a point. In the real world, you just track how they go. So how we do it is something on the lines of swimming. So if you are swimming straight, you beat both your arms equally hard. But let's say you're trying to swim right, then you would beat your left hand harder than the right. So you'll push more on the left than the right, so you end up turning right. And if you want to turn left, you push more on the right versus the left, which is what the fly does. The fly is swimming in a fluid called air using its hands called wings. So it's flying here, and what I do is I track the edges of the wing in real time. So there's a camera which takes the video of the fly rapidly, 100 times a second, and every time it takes an image from the video, it tracks, okay, it wants to take left or turn right. Oh, the video says, oh, it has to turn 30 degrees left. And so what I do is this, I have two choices. The fly wants to turn 30 degrees left. So I can either turn the fly 30 degrees left, but really in the real world there are no motors which can move so fast. You can't move things so rapidly as fast as flies. So what we do, we move the images on the monitor 30 degrees right. Like when you're going forward in a train, you feel like you're going forward because the images of the sky and the grass and the ground go backwards. So in this you can see when the fly is going forward, everything else seems like it's going backwards. And if the fly wants to turn left, I would move the images on the world right, like this. Right now it's the fly who's controlling the world. It's looking at the grass, it's looking at the sky and saying where it wants to go. And you can also see its trajectory here, which is the virtual path the fly has taken. It's now going in circles because there's nothing much to go. If I change the settings a moment, you will see something nice. So you can see the grass, you can see the sky. This is the trajectory of the fly. So in this world there is nothing, there is no trees, it's just the empty land world which is just grass and sky. They're going all directions. Now you see there's a tree there and they go straight to the tree. You can see there's a tree and they're fixating on it and they're going to the tree there. They go in circles, there's like it's going away, now it came to the tree, it's going to it. It's sort of very not precise is because they're sort of, if I have to use the words, distracted by the clouds in the sky or the patterns of the clouds in the sky. So if I now turn off the clouds, which I might say in the real world slightly tricky because you can't just say, okay, all the clouds become invisible right now, which is why we use the VR. Right now I can just turn off the clouds and the clouds go poof and there's no more clouds. It's a blank sky, like I said, it's overcast. And now you can see that it will go to the tree much more easily. So you can see it goes straight to the tree without issues. You can see it right now, it's going like straight like an arrow without any issue. And you can see the trajectories here too. It's going all over the place in the previous trajectory and now it goes straight like an arrow. Basically flies can see patterns in the clouds. We can also say other things, but these are the few things which you can do easily in the virtual reality arena, which is sort of nearly impossible in the real world.
Great job, man.
So I can also show you, I can't show you this part which is giving it order, not because it's secret, because I've been trying it since 3 months and they've not really flown sensibly because I don't know what's wrong. They seem to go downwind. So I can't show it because I don't know what they're doing myself. So I really can't say a solid conclusion. I can say this, that if I put the tree further and further away, beyond a point they don't find the tree. Makes sense. Like the tree became so tiny they don't find it. But what I'm saying is I can tell you that for this fly, a tree which is 20 meters away is nearly invisible and they will never go there. Imagine doing that in the real world. Doing that experiment is really hard. You really can't track the fly. In this, I can track a 7 mm fly and tell you that 20 meter far away tree is nearly invincible, suggesting they should use odor to find it, which is my next research step. But I don't know how do I conclude it, but if you have a particular way, I can end on that. But I can just say you can learn a lot with this technique which is sort of impossible to do in the real world.
That sounds like a good ending. That was a great visit. Thank you for that, Shannon. And we're back to where we started from.
Yes.
So your entire research is extremely fascinating and
Thank you.
Makes me wonder, what's the vision? Where do you see yourself with the research in the next 5 or 10 years?
So I would say that, the question that we ask, how do animals identify objects? It's such a huge question. I actually almost think it's a question that can't really be answered. but that's not really the point. Of course, we're trying to answer the question, but I think that it's more important the journey that we go on, and the experiences that we have, and the new tools we developed, and the new concepts that we gain knowledge of through this process. So for me, it's much more about the journey of the science than actually getting to the end point. That's important. So I have trouble telling you where I'm going to be in 10 years. I don't really know. We go where the research takes us. I didn't think I'd be working on pollution a few years ago. If you'd have asked me, I would have said, why would I be working on pollution? But it became necessary and important to me once I came to this country and I saw how evidence of pollution affecting the very things I work with. So I don't know where I'll be, but I hope I will be doing science, and I hope we'll still be continuing to answer questions, and I mostly hope that I continue to have such fascinating and wonderful people to work with. I'm so proud of the lab I'm part of. It's a group effort, and we are collaborators with each other, and they are as much a part of the NICE lab and the NICE idea as I am. So it's really a collective vision that we have.
And what would be your message to the budding Indian scientists?
My advice would be to not be afraid to ask questions, to not be afraid to be curious, and not be afraid to question things, even things you read about in a textbook. Wonder why is that? Not just take it for granted that this is the way DNA functions or this is the parts of a cell, but wonder why are they there? Because science is changing every day, and every day we learn something, and we maybe overturn an idea that people had thought was correct for so many years. So I would say be curious about the world around you. Our inspiration comes from the marketplace. Our inspiration comes from everyday life that we have around us, just asking questions. So I would encourage scientists, not just in India but everywhere, to do this, but particularly in India. I would say that there's a lot of problems that India has, and India is in many ways a microcosm for the rest of the planet. So I would encourage young scientists or people wanting to go into science to think about how they can actually try to solve some of these problems with water usage or natural resources in general, deforestation, disease prevention, food security, urbanization, you name it, it's all happening here in India, and we need intelligent, bright, curious people to do this. And I think if they do that, they will be able to solve problems not just for India but for this entire planet.
Well, thanks a lot, Shannon, for your time. It was extremely enlightening talking to you, and I hope so it's for my viewers as well. Thank you.